All right, so my talk will be about immunoglobulin, heavy chain constantine, polymorphisms, and why we believe that some of these polymorphisms might be of relevance to multiple sclerosis. So I'll start off by introducing you to the disease multiple sclerosis and the importance of B cells in this disease. Then I'll take you on a slight detour to recapitulate some important points about the polymorphisms of the IGKV1 gene, which encodes the IgG1 isotype of the immunoglobulin heavy constant chain. Next, I will explain why we believe that some of these polymorphisms might be of importance in multiple sclerosis, and I will show you some data from our results to convince you of this. And finally, I'll present one of the current projects that we are working on, where we decided to go all germline and do ultra-long sequencing using Oxford nanopore technology. So, multiple sclerosis.
Is anyone able to assist with the audio?
Pardon? All right. Thank you. MS is a chronic inflammatory disease in which the immune system seems to attack the brain and the spinal cord. And the primary target is believed to be the myelin sheets covering the axons. But in fact, it also seems to be a destruction of the axons themselves. Correlates much better with the disability of the patients. And the symptoms of MS reflect the parts of the brain that is affected. And typical symptoms include reduced vision if the optic nerve is affected, or a tingling sensation in an arm, a leg, double vision, dizziness, and sometimes partial paralysis. And a typical course of these symptoms is that they last for some days, weeks, and then they gradually subside without therapy. Now, this is a blood vessel, and this is the brain. And in MS, both innate immune cells, such as macrophages, and adaptive immune cells, T cells, traverse the blood-brain barrier. They move into the brain where they damage myelin sheets and axons in different ways. However, B cells seems to be playing a crucial role here. And one of the most compelling arguments for this role of B cells, for this key role of B cells, is that the most efficient drugs that we have today to treat MS is targeting the surface molecule on B cells. We're using monoclonal antibodies to target CD20. And these drugs very efficiently suppress the inflammation in MS. This was a surprise the first time they tried this almost 20 years ago, as MS was thought to be primarily a T-cell mediated disease. And ironically, monoclonals targeting T cells did not have much effect. So, B cells have been known to play a role in MS for quite some time now. And as part of the diagnostic work of MS, individuals that are referred to the Department of Neurology undergo diagnostic lumbar puncture, where we are sampling a small part of the cerebrospinal fluid, which is a fluid that are covering the brain and the spinal cord. And within this cerebrospinal fluid, these individuals have antibody-secreting cells that are producing immunoglobulins, mainly immunoglobulin G of the IgG1 isotype. And the most sensitive way to detect these immunoglobulins is to perform a technique called isoelectric focusing, which is a type of electrophoresis, which then shows that there are IgG bands that are present in the cerebrospinal fluid that are not present in the serum of these individuals, which indicates that these immunoglobulins are produced intrathecally in the cerebrospinal fluid and probably also in the meninges of these patients. Now, although this is used as part of the diagnostic criteria of MS, it's not specific for the disease. We see these IgG bands also in other diseases, such as other neuroinflammatory diseases, and the largest group is actually chronic infections of the CNS, such as Lyme neuroborreliosis or herpes encephalitis. However, in such cases, the IgG is directed towards the causative antigen, such as the Borrelia spiral jet. In MS, on the other hand, we do not know what the target antigens are, and there is no current consensus on what the T cells and the B cells react to in MS. And although the list of publications revealing novel antigens is quite long, and I put just some of them here, these candidates have not been validated by independent research groups, and some of these candidates are also controversial, I must say. So if you're interested in that, come by and we can discuss them, but we will not dive into that topic now. So now we will leave MS for a couple of minutes, and we will take a slight detour to take a quick recap of what is known as IGHG polymorphisms and allelic exclusion cells. Important to give you some context in order for you to understand the results. So there is an increasing number of IGHG1 alleles that are being characterized, and these are only a small number of them. It's also from the IMDT database. Most of these alleles, however, carry silent polymorphisms that do not lead to any changes in the amino acid sequence of the protein. There are important exceptions, and these exceptions involve amino acid changes known as allotypes. And the allotypes of the IgG1 molecule are located in the CH1 and the CH3 region of IgG1. And they are called G1M17, G1M1, and G1M3. There are also a couple of other allotypes that have been discovered, such as G1M2, 27, and 28, but I will leave them out for now, for simplicity, because it's not important to understand this story. In individuals of European descent, the same set of allotypes in the CH3 region are always inherited together with the same allotypes in the CH1 region. So these alleles can be grouped in two, where one of the groups are encoding G1M17-1, and the other group of alleles are encoding G1M3. However, the two most frequent alleles encoding them in the population are the O2 and the O3 alleles. I also want to just quickly remind you of the principle of allelic exclusion in B cells, which occurs during the VDJ recombination in the bone marrow. Now, these bars symbolize the kappa and lambda light chain genes and the heavy chain genes. And we inherit one copy of these genes from our mother and one copy from our father. However, during the VDJ recombination process, only one set of light chain genes and one set of heavy chain genes will be used. So when a mature B cells leaves the bone marrow, it will use 1 out of 4 possible light chains and 1 out of 2 possible heavy chains. And a biological rationale for this could be that we want a certain B cell to target one antigen. However, some of you might know that reality is more complex. We do find a lot of B cells with especially several light chains out there. But this is at least the main rule. So if you look at the approximate distribution of the IgG1 allotypes in individuals of European ancestry, around 40% will be homozygous for M3 alleles, around 10% will be homozygous for M1 alleles, and around 50% will be heterozygous for the M3 and M1 alleles. And due to the random allelic exclusion in the B cells, if we zoom in on the heterozygous individuals, they will in essence have two different IgG1 B cell populations, where one B cell population carry the M1 version of the receptor and the other B cell population carry the M3 version of the receptor. So what does all this have to do with multiple sclerosis? This story begins a long time ago, in 1981, which was the same year as AIDS was described first time actually, and the same year that Prince Charles married Lady Diana. And this paper was published in Science, where Salier and Handy showed that if you measured the concentration of IgG1 using antibodies against the M1 and M3 allotypes, you saw that there was more M1 than M3 in the cerebrospinal fluid of MS patients, in M1 and M3 heterozygous MS patients. Okay, a priori we would believe that in heterozygous individuals that carry both M1 and M3 alleles, you would find approximately 50% M1 and 50% M3 immunoglobulin. This was not the case for heterozygous MS patients. More recently, research groups have looked at the amount of IgG that is found, the levels of IgG in the cerebrospinal fluid. And they use a measure that is what is called the IgG index. And it's shown that there are high levels of IgG in patients that are carrying the M1, either if you're homozygous or heterozygous for the M1 allele, you have higher levels of IgG1. There is also one genetic study that show that your IGHV1 haplotype might actually influence your risk of having MS. Although this study needs to be reproduced. Also based on what we know today about the complexity in this. In relation to the allotypes and MS begins with an argument I had with one of my former supervisors, Frode Vartdal. And Frode and I did not agree on how to interpret this old science paper. So in my opinion, and I'm still of that opinion, it's more intuitive to imagine that these allotypes, these amino acid changes in the constant region, could affect binding to Fc receptors. And that the result of this bias, D101-3 bias, comes from the distribution of IgG within the central nervous system. For instance, we know that the neonatal Fc receptor is abundantly expressed in the blood-brain barrier. Frode, on the other hand, he claimed that this might actually be due to the cells being selected into the central nervous system. That cells carrying M1 B cell receptor is selected into the CNS. So of course we needed to check this, and we used antibodies against these allotypes, and I labeled them. And I did intracellular flow cytometry, and I looked at the B cells. And I gated on antibody-secreting cells, and I gated on IgG1. And the IgG1 population was again gated into the M1 and the M3 allotypes. And this is just one example where you see that in blood we find approximately 50-50 distribution, which is expected. This is one heterozygous individual. It's expected to have a 50-50 distribution of these two alleles. However, in the cerebrospinal fluid, there was this smack you in the eye thing that he was right. This is actually the B cells that are carrying the M1 that is selected to the cerebrospinal fluid of heterozygous patient. And this is the summary graph, which shows that it's always towards the M1 allotype. And the skewing is quite pronounced. So then we asked, has this something to do with MS, or is it something that we can find in your inflammation in general? So we wanted to include the control group with neuroinflammation unrelated to MS. And we wanted to use chronic infections. So we included patients with Lyme neuroborreliosis, but it's very hard to get the fresh cell samples from these patients because you need to be there at the department the moment they are diagnosed. So we found a way around that. We used biobank samples, and we didn't look at cells, but we looked at the IgG. So what we did was isoelectric focusing, and looked at the IgG that is being produced in the cerebrospinal fluid. And this is one example where you see that when we stain for antibodies against M1 and M3, there are bands of both allotypes present in patients with Lyme neuroborreliosis. But there are only bands here that are stained with M1 in MS patients. And this is the summary graph of 16 patients with neuroborreliosis and 28 patients with MS, which is pretty strong evidence, in my opinion, that this phenomenon is present in MS, but not in Lyme neuroborreliosis. So we wanted to explore the phenotype of these cells using single-cell RNA sequencing, and we sorted single cells based on markers for antibody-secreting cells, and we also sorted all other types of B cells were CD19 positive, and we used a single-cell sequencing RNA sequencing protocol, which is called SmartSeq2. And the advantage of this protocol compared with text, for instance, is that you get the full-length mRNA molecule. And this, by this advantage, we could do transcriptomic profiling, and we could also custom these molecules, the immunoglobulin molecules, back together to reconstruct the complete cell receptor with a paired heavy light chain. Now, this is the work of Ida Lindemann and Justina Olak. And Ida Lindemann was instrumental in analyzing these data, and she had been Sanger Institute, and she had wrote a pipeline with Mike Stubbington, which is known as Bracer, which is enabled us to test these things back together. So these are UMaps showing unsupervised clustering of the cells, and the antibody-secreting cells are the blue here, clustering separately from all other B cells. And if we zoom in on these other B cells, we see that the naive cells are clustering differently than the memory cells, which also follows, I guess, that there are some clustering here according to the isotype. If we look at the naive isotype, they cluster differently. However, disappointingly, there were no obvious clustering according to the allotype here, indicating that the M1 and M3 cells do have a similar phenotype. When we looked at the allotype of these cells, we did find the same skewing, although maybe not that big. This is antibody-secreting cells to the right and memory B cells to the left. See that you see this skewing among the antibody-secreting cells, but not as strong among the memory B cells. However, what we also observed and what we found intriguing is that if we stratified the patients and the cells according to allele type, here you have the M1, here you have the M3 patients, it seems that patients with M1 alleles show kind of a stereotype pattern here. They overuse certain variable genes of the IGHV4 family, and they overuse kappa light chain compared to the individuals carrying the M3 alleles. This is how it looks like if we pair the heavy and light chains. Seems to be certain combinations down here of IGHV4 family genes pairing with IGKV1, which is, this pattern seems to be present in M1 but not, at least not that degree in M3. And the pattern is more chaotic if we break it down to specific genes in this circle spot, but there is no representation of certain combinations, such as IGHV439 with this one here, IGKV1-5. All right. So this observation that it seems that it could be a possible connection between a heavy chain constant region alleles and certain variable genes. Together with the realization that we may not know enough about this immunoglobulin constant region to generate sound hypothesis on the phenomenon that we observe, has made us go into germline sequencing. And so finally, I'll take you through some of our efforts to use the Oxford nanopore sequencing to assemble and paste the IGH locus. So this is the work of Mari Gornitska and Egil Røsje in my lab. And we do this in collaboration with Corey Watson and Udalok Jana in his lab, where we validate our assemblies using PacBio circular consensus sequencing reads. And Mari is currently writing a methods paper on this, where we try to benchmark this pipeline. So our pipeline works with we started off using isolated monocytes, but we now see that it works as well with PBMCs. However, EBV-LCLs may contain deletions of parts of the locus of one of the both haplotypes and are more difficult to assemble. So we used a specifically designed protocol to extract high molecular weight DNA. The genomic DNA is sequenced on a nanopore Promethean sequencer. And nanopore sequencing works that way that the DNA is moved through a pore, creating differences in electric current that is interpreted as different bases. And we also use an option that is known as adaptive sampling. And this is a possibility with nanopore that you can feed the computer with your reference of choice, in our case, an extended IGH reference. And the pores will read this in real time. And if it's not in our region of interest, it will spit out the DNA from the pore that does not match. And by this, we are able to enrich for reads mapping to IGH by a factor of 30 to 40 ampere. So this approach yields an N50 of more than 65,000 bases, and we are generating one contig per haplotype, covering the entire locus of more than 1 million bases. Luckily, the PAC bioreads from Corey's lab seems to agree with our assemblies. So in this figure, this is 2 MS patients where Mari has mapped the PAC bioreads back to our assemblies, and then she has used a variant caller to see if there are any differences between PAC bioreads and our assemblies. And you see, if you look for the high quality differences, the Q-score above 20, there are no differences. There are a few differences of lower quality here. But considering that this is 2 haplotypes covering 2 to 3 million bases, it's not a lot. So in this methods paper, we sequenced 4 individuals, and we discovered one large duplication of the constant region of one individual. And what we believe is a deletion of the entire IGH locus of another individual. So there are large structural variants out there to be discovered. And why do we believe that this can help us with what we are struggling with in MS? Well, we cannot be sure of this, but we felt we needed to take a step back to characterize this especially the constant region better, to be able to interpret our findings in MS. And we believe that when we get more data, if we get enough data, we will be able to see if there are any linkage, disequilibrium between the variable and the constant region that could explain some of our previous observations. And secondly, it might be in the future, through our data or also through data generated by other labs, such as Corey Watson's lab, possible to create pan-genomes of representative populations to perform genome-wide association studies of the IGH locus in MS, which I think would have been very cool to do. So finally, I would like to acknowledge all previous and current co-workers on this project and the collaborators that have and are contributed to this. Thanks. Are there differences in glycosylation patterns between the 2 alleles? Between, there are differences in glycosylation patterns if you look at the IgG1 that we find in MS patients compared to other population or other patients group and healthy individuals. But as far as I know, I think these allotypes are not one of these amino acids that are being involved in the glycosylation. So they are not involved in N or O glycosylation, as far as I remember. So I don't expect that this would be the case. But we haven't checked. I mean, it could be differences other places, not related to the location of the allotypes, but we haven't checked.
So what? Oh my God! What mechanisms would you speculate if not something like glycosylation? There's questions about class switch recombination, but
yeah,
what would you think is going on?
I don't know. If you have any, if you have a good suggestion, that's sort of what we wanted to take a step back and go all germline as well, just to see what's actually there, because it's very hard to interpret if you don't have the map. But of course, one could speculate because. One could speculate around FC receptors, but I don't see why that can explain everything, because binding to FC receptors will always be downstream, right, of the selection of the B cells. Yeah, one thing that have been proposed is indeed the class switching, that there could be some cytokines that are, for some reason, having higher affinity for B cells, M1 B cells. Yeah.
Great. I think we're out of time, but I noticed that there's some very nanopore specific questions up there, so maybe you guys can find Andreas. Never mind. The president told me that we can keep going. Let's do one more. He's in charge. I'm just.
Quick answers.
So tell us about the nanopore, because I think that's a hot topic.
What's the question?
It's gone.
Okay.
I don't know what it was. Can we go to the top? There we go. Was adaptive sampling better than lab-based enrichment in terms of enrichment?
Oh, we haven't tried lab-based enrichment. Good question. We probably should try that.
Let's see. You entered CSR. You sort of talked about speculating around FC receptor, but I suppose you haven't directly examined genetic variation in the FC receptor genes. Is that true?
Others have done that, and it doesn't really correlate.
Cool. Good. Are you good, Victor? All right. Thanks, Andreas. That was great.